actually. Hi, everyone. And you need this. Can you guys hear me? Of course, I can't hear you.
Yeah, we can hear you, Abby.
Okay, great. And let's see. All right, and you're managing the participants, right?
Yeah.
Okay. I don't know how this is going to go because I'm a little more disorganized, as you can tell, than usual. And this is also a topic that is, it's a little bit newer in terms of our understanding of it, and it's not really in the book, except in passing. I mentioned on the syllabus, there's just like 2 pages in the book that kind of talk about this phenomenon of intracellular phase separation. But I want to talk about it because I think it's very cool and important. And if you are interested in cell biology, it's something that is sort of a past and future topic. The importance of phase separation within cells is something that was very clear in the mid-20th century. And then I think what happened is that as molecular biology grew, sorry, I'm going to turn off that chime, as the era of molecular biology enabled researchers in the late 20th century to start looking at individual components within cells, they actually started to forget about the physical properties of things within cells and we have, you look at textbooks and you see these images of cells that are very empty looking. You have just like a shell and a few sparse proteins. And one reason I keep using these images from David Goodsell is because his illustrations are really nice reminders that cells are actually extremely crowded and heterogeneous environments. The interior of cells is not only filled with organelles that physically separate different processes, but as I'll talk about today, we're now reappreciating that even not thinking about membranes, there are many other mechanisms that concentrate components within cells and create what we now call condensates or phase separated bodies. And without getting too detailed about sort of the theory of how these things happen, I want to talk a little bit about a couple of examples of bodies that have been studied recently and a little bit about what we think about how they form. Okay, so I actually kind of touched on this. I sort of skipped this part in talking about nuclei. We talked about chromosome architecture and the nuclear lamina and the nuclear envelope and nuclear pores. And I left off with just a quick mention that nuclei are actually filled with a bunch of different little bodies that were sort of discovered by making antibodies to proteins that people were studying and using them to stain cells. And many of these antibodies wound up lighting up little globules inside the nucleus. And some of them got names for people who discovered them or some protein that was inside them or something like that. The biggest nuclear body, other than the nucleus itself, is this kind of blob that's often in the middle of the nucleus, but it can be anywhere, called the nucleolus. And there can be just one nucleolus in a nucleus, or there can be more than one. It depends on a few things, which we can talk about if you're interested. But it's this very prominent body that fills often a substantial volume within the nucleus. And unlike the nucleus itself or other organelles, it does not have membranes around it. It's a pretty irregular shape. Often, in this case, you can see 2 of them in this image, and it's just this darkly staining kind of heterogeneous material that was seen by electron microscopy. You can even see it by light microscopy because it has a different refractive index than the surrounding material, so you can often see it by phase contrast microscopy, and I'll show you an example of that. So we've known for a long time that these bodies within the nucleus have a special function, and that function is to do part of the process of ribosome assembly. So ribosomes make up a lot of the mass of cells, right? They're very large, complex machines that do protein translation. And they are made of both RNA and proteins, many proteins that are arranged around a scaffold that's formed of a couple of large RNAs. And those RNAs, like all RNAs, are transcribed in the nucleus, right? So there are chromosome regions that have genes on them that encode these RNAs that are not ever going to be made into protein. They are just, they're structural RNAs, and their job is to kind of fold up and recruit a whole bunch of proteins that bind to specific little loop structures or other binding sites on the RNAs. And so the RNAs act as a scaffold to bring together all these proteins. And some of the proteins start assembling on the RNAs while the RNAs are still in the nucleus. And then the process is actually complicated. So this shows a kind of cartoon view of it. But here's the RNA being synthesized at its site from the chromosome inside the nucleus. It happens in this case, I mean, we don't really talk about transcription in this class, but these large RNAs are actually synthesized by RNA polymerase 1, not RNA polymerase 2, which is kind of the main RNA that makes protein coding gene RNAs. So these long RNAs are made in the nucleus, and they start to recruit a bunch of these proteins. And the regions within the nucleus where that is happening are the nucleolus. And so this is all the nucleolus. And partially assembled ribosomes actually then get exported through the nuclear pores and undergo cytoplasmic maturation. So the assembly of ribosomes happens, some of the proteins are made in the cytoplasm and imported into the nucleus, where they go into the nucleolus and assemble into these pre-ribosomal subunits, and then the ribosomal subunits come out of the nucleus back into the cytoplasm. So these sites of active ribosome assembly inside the nucleus are these bodies called nucleoli, and they're nucleated at the site where the RNAs are transcribed, the long RNAs. And so in some organisms, the RNAs are localized to one or a couple of chromosomes. In some genomes, actually, there are genes encoding these ribosomal RNAs on multiple chromosomes. And what often happens is that those regions of the genome, all these different chromosomal regions, actually come together and sort of, they don't meld together, but they all get sort of sucked into this nucleolus because they're all contributing RNAs to this ribosome assembly process. Okay, so that was known. I mean, we've known this for a long time, and people have observed that even within the nucleolus that there are these different sort of appearances, that there's material that's called the granular component and the dense fibrillar component and so forth. There's actually at least 3 different kind of appearances of different regions within in the nucleolus. And that's just kind of another representation of what I already showed you, that different chromosome regions converge there. And what people have sort of realized recently is that these nucleoli are actually liquid. And this is a way that people test whether something is liquid within cells. Here they're taking, this is a xenopus oocyte, so it's a very, very large cell, and there are actually many nucleoli in the nucleus of a xenopus oocyte. And these cells are so large that you can physically manipulate them. So here the researchers are going in with a glass needle, and they're pushing together 2 nucleoli, and what they see is that they will actually fuse together. And that behavior of fusion is characteristic of a liquid. So if these things were kind of gel or solid balls, they wouldn't undergo the kind of shape changes that are needed to take two balls and fuse them to make a single ball. And here you can also see the heterogeneity of the nucleolus. First of all, you can see the nucleoli. This is I think, phase contrast imaging. And the reason you can see the edge of the nucleoli is because these bodies actually have a different refractive index than the surrounding nucleoplasm. And so you can see the edge because it changes, the light there changes speed. And you can also see the heterogeneity of the nucleoli as little balls within the bigger balls. So those are actually subregions of the nucleolus. And I'll show you another view of that in a minute. All right, so this is another, this is from the same paper. I showed you an example of nucleolar fusion, and what the authors also found that you could basically cause these bodies to separate, to undergo a fission process by applying shear stress. And again, that is a property of a liquid, and the way that this happens, like you get this kind of bridge, and then eventually this bridge kind of gets resorbed into the droplet on either side. And that resorption happens because there's a drive to minimize the surface of this, just like, well, we'll come back to that, but there's a energy cost associated with this kind of surface, and so the system wants to minimize that energy by making each blob as round as possible, so minimizing the surface area to volume ratio. So you're familiar with this in everyday life, right? This is, I can't see myself right now, unfortunately, but I wish I could, but maybe I can try to turn this on. Skip it, not worth it. So, okay, this is just a bottle of water and oil, just cooking oil. I dyed the water blue just by putting some food coloring in it, so it'd be more obvious. And I mean, when you make salad dressing, you shake it up, and what you're looking at actually are droplets of one liquid inside the other. And sometimes, if you shake it enough, you'll get droplets of oil in the water and droplets of water in the oil. And if you shake it enough, you may even be able to get them to almost completely disperse or make the droplets so small that you effectively get an emulsion. This is kind of an emulsion that I made by adding some soap to this mixture of water and oil. So now that they haven't completely fused together, there's still two different liquids in here, but the surface tension with the soap in here is much lower. And so there's less of a drive for these things to completely separate and to minimize the surface tension by forming just one oil and one water phase. So I'll come back to that, but maybe keep that in mind as we're talking about this. Okay, so I mentioned that this idea that there are kind of liquid subcompartments within cells that are not surrounded by membranes, in addition to the ones that are surrounded by membranes, has actually been around for a very long time. This is maybe the oldest mention I could find of it. This is a paper from the late 19th century. You can tell they didn't have cameras. They were just drawing what they saw in the microscope, or in this case, it's a single author paper. And he described the behavior of these nucleoli, which he was observing under the microscope, as liquid-like. And he described it as sometimes having a viscid or thick consistency. And in other cases, it's more fluid, and thus it attains a more spherical shape. And it behaves like a thin liquid, a non-viscous liquid. And it was, I mean, in this paper, he pointed out that this fluid-like behavior probably allows the nucleolus to attain all these different shapes and configurations. Sometimes you see many, sometimes they all fuse together. And he also kind of speculated that this enables the nucleolus to divide as cells undergo division. So this idea has been around a long time. So I'm talking about liquids, and I'm actually going to try this poll feature. I don't know if it's going to work. And how do I launch a poll? Polling. I'm going to try to launch this as a poll. So see if you can answer this, and I don't know what's going to happen now. So this is a multiple choice, and just click on the feature or features that you think define liquids. And I'll give you, like, I don't know, 20 or 30 seconds to think about this. By the way, this is anonymous. I'm not going to keep track of which answer you wrote, and I don't even know. Okay. So,
just out of curiosity, can students see the other students' answers? Because I can see everyone's answers, but I don't know if that's because I'm a co-host.
I can see. Can you see who's answering?
No, we can't see who's answering. We just see like the number of people answering each thing.
Okay.
Oh yeah, I meant the number of people. Yeah. Okay. good to know. I just wanted to make sure people weren't being influenced by other people's answers.
That's a good question. Okay, so that's been a minute, so I'm going to stop it. And actually, you guys did really well. So 45% of the people who answered said that molecular motion of molecules is a feature of liquids, and 45% also said ability to change shape. And those are both right. So the molecular motion of molecules within a substance is a defining feature of liquids. The thing that makes liquids able to take on the shape of a container, or to undergo rapid fusion or fission, where the droplets change in their topology, in their shape, is the fact that the molecules within the liquid are weakly bonded to each other. So strong molecular bonds are a property of solids. When things are tightly bound together, then those bonds don't easily break and reform. And so that makes something a solid, because it doesn't tend to undergo shape change. But within a liquid, and it doesn't have to be an aqueous liquid, oil is also, some oils are liquids, so they're not hydrophilic, but they have molecules that are weakly bound together that enable the oil to also have this fluid property. So when we're talking about liquids within cells, we're talking about condensates, or concentrated balls of molecules that they are not, it's not quite like oil and water, because these condensates, for the most part, do not exclude, they don't exclude water. So it's different from, so we've already talked about one kind of condensate, which is, sorry, I'll come back to that, which is a membrane itself, right? Membranes are made of liquids. Membranes are made of lipids. Lipids do not like to mix with water, and so they phase separate to form a separate material within the cell that actually excludes water. But the condensates that I'm talking about today are a little bit different. They don't hate water nearly as much as lipids do, but they tend to be, the molecules that are in them tend to, they don't like water as much as they might, and they like each other. And so they come together because it winds up that the cost of coming together, which reduces entropy, is compensated by the molecular bonds that form within that material, giving the system enthalpy. So there's kind of a trade-off, and I'll try to talk more about that. Okay, so this video I think is kind of helpful, although it seems a little bit off topic.
Pouring fire ants through a funnel is not something you should try at home. Nobody wants that many fire ants in their home. But for researchers interested in the physics of fluids, pouring fire ants through a funnel because
Sorry, it's playing twice and I don't know why.
Yeah, we weren't able to see it initially. Now I think we can see it.
Yeah, I think it just didn't have an image initially.
Huh.
Pouring fire ants through a funnel is not something you should try at home. Nobody wants that many fire ants in their home. But for researchers interested in the physics of fluids, it's irresistible because they
Abby, I think we still aren't able to see it.
Fluid and a solid.
I think this is a Zoom
See
problem.
how the ants drip like some kind of syrup? But then, watch as
I wonder if it's possible there's a copyright.
It springs
I
back.
don't think so, because I actually downloaded
solid,
it.
like rubber.
I think it's a Zoom.
That's a rare combination, and nobody had seen a group of living things behave that way before.
So frustrating.
Now, the researchers didn't just poke at the ants and
either.
pour them out of tea kettles and put them through funnels. They did serious ant ball physics with lab devices that tested the viscosity and the springiness of it.
All right, I'm going to skip that. Am I still online? Sorry about that. I will send you the link. Anyway, it's very cool. And what it shows is that ants stick together, and they make like rafts that can sort of float on water. And that's actually how fire ants move around. And I know that my slides have closed, and I'm trying to reopen them.
Maybe you can just share us the link and then we can look at it later.
That's what I'll do.
Okay.
Uh-oh. I also have a terrible feeling that I did not, may not have saved the most recent version of this, so we'll see. Okay, so I think we're okay. All right, so I'm going to move on. You're probably not. Can you see my screen? Can you see my slide?
Not right now.
Oh, okay. This is not my day. All right. Can't find the screen sharing or the Zoom controls in general. Okay.
Able to see where the ID is?
Yeah.
Okay. Okay, we can see it now. Great.
So sorry, guys. Thanks for your patience. Okay, so this just highlights that I kind of touched on this already, that nucleoli look like a droplet, but actually if you look closely by phase contrast, or if you attach different fluorescent proteins to different components within the nucleolus, you can actually see that it's a heterogeneous liquid. There are multiple liquid phases within the nucleolus, and this paper just talks about the fact that they correspond to these regions that were previously identified based on their sort of appearance by electron microscopy of the granular component and the dense fibrillar component, and I think it's just fibrillar component. So different functions are known to be associated with different proteins, and we can see that they are physically compartmentalized within the nucleolus, and it's likely that this kind of spatial partitioning of the nucleolus allows for an efficient sort of bucket brigade of ribosome assembly as components move from one component to another component to do successive stages in this assembly process. Okay, so I still haven't really talked about what is a condensate and how does it form. And this is, as I've alluded to, this is a very common phenomenon within cells. And what we now understand, and actually there's theory going back to the mid-20th century from polymer chemistry, is that when you have a solution of certain kinds of macromolecules, and so you can think of a cell as a solution of macromolecules, some of those macromolecules may like each other and may not like the liquid very much, and they can undergo this process of condensation, or it's often called demixing, to form a condensed phase and a dilute phase. So the condensed phase is here. It's not excluding the solvent, so there's still water in here. It's just there's a higher concentration of these molecules, and they're bound together by weak molecular bonds, and that's what makes this a liquid.
Sorry, so you said the one on the right is the condensed phase?
This is this purple cluster with the bonds between the purple molecules is the condensed phase, and what's surrounding it is the dilute phase, where you still have some of these purple molecules. They're not bound together. They're solvated. They are completely surrounded by the solvent, but here they're making interactions with each other to form a condensed phase. And typically, a lot of these phases within cells are very dynamic. So a little change in how much these purple molecules like each other or how much they like the solvent can switch the system from being fully solvated or fully, just a single phase to a two-phase system where you have a dilute and a condensed phase. Okay, I know a lot of these concepts are new, and I'm trying to say them in multiple ways because people describe this in multiple ways in the literature, so you may encounter kind of different descriptions. So are you with me so far? I'm gonna
I actually have a question, if you don't mind.
Oh,
the one on the right, how would you define the right image compared to the left image?
how would I describe this?
Like what would the vocabulary be when you have a greater concentration of aggregates?
So this would be a single phase system, and this would be a two-phase system. With respect to the purple molecule. Okay, so in this case, the purple molecule is in a single phase. It's at the same concentration throughout the system. And here, it's at two different concentrations. Within these condensates, it's more concentrated, but there are also still purple molecules that are fully solvated. There are just less of them than in this case. And that's the dilute phase. Does that make sense?
Yeah.
Okay, I know it's kind of confusing, and we're often talking about systems that have more than just single molecules involved, but in this case, the blue is just the solvent, which is generally water in biology. Okay. I know this is weird. So, okay, so here's kind of an animation that I made where you have, in this case, you have like a yellow protein that's fully, it's fully dissolved. It's completely dispersed in a single phase system. But what can happen is that the protein gets modified and either likes itself more or likes the water less, and that drives it to form a condensed phase. And so you see that it forms multiple droplets, and those can start fusing together to form, just like oil and water will tend to completely separate. If this persists long enough, these droplets will potentially fuse together. There's also another way that the systems can, the tendency will be to go toward a single large droplet of this yellow stuff in the surrounding blue stuff, because again, that minimizes the what's called the interfacial surface. I mean, you know that we talk about surface tension, which is the tension between a liquid and the air. When you have a two liquid system, the interface between the liquids has an interfacial tension. And in the case of oil and water, it tends to be a very high interfacial tension because these liquids really dislike each other. But in a biological condensate, the interfacial surface tension tends to be lower because these proteins or macromolecules that are concentrating in here don't hate water. They just don't, they like each other enough, and they form bonds with each other enough that the bond formation offsets the loss in entropy caused by concentrating them within a smaller domain of the system. Okay. All right, so I've already used a bunch of different kind of terms, and different people have different ways of describing this phenomenon within cells. So I just kind of made a little list here of terms that people use to describe this phenomenon. And this is, yeah, so this specifically applies, these terms specifically apply to cases where you have separation between a dilute phase and a condensed phase, and usually the condensed phase is still liquid. It doesn't have to be. The condensed phase can sometimes become more solid or gel-like, as I'll touch on toward the end of the lecture. But most of the cases that we're talking about now, the condensates are liquid. And those condensates are sometimes called biomolecular condensates or just condensates. Sometimes they're called membraneless organelles. A technical term from chemistry is coacervate, which is a particular kind of chemical attraction between oppositely charged molecules, but sometimes these are described as coacervates. And most generically, I think a lot of people describe these as phase-separated compartments. And again, you have like a condensed phase and a dilute phase. And because this area of cell biology is sort of undergoing a rebirth in the last 10 years or so, it's undergone a major rebirth, and a lot of people are sort of still getting educated about what is happening at a biophysical and chemical level. Sometimes people use weird terminology that may not be strictly correct. And so I'm going to try to avoid that, but you may encounter different ways of people may describe these phenomena in different ways, which may seem incompatible. Okay, so I've so far just talked about nucleoli and the fact that they have liquid-like properties, but actually there was a paper just over 10 years ago that really brought this whole idea of intracellular phase separation into focus, and it was about these bodies that are called P granules or germ granules, which are present in many organisms. Oops, and I see my slides are out of order. So I'm going to skip this for a sec. I'll come back to that because nuclear pores are also relevant here. So P granules, which were discovered in C. elegans, but also exist in basically all animals, are these, they were called granules because they look like little dots or little bodies. And these are images of embryos from C. elegans, from nematodes. And these P granules they are known to contain a whole bunch of RNAs for genes that are supposed to be expressed in a highly regulated way in the germline. And they also contain a bunch of proteins, the first of which was called PGL1 for P granule 1, that bind to the RNAs. And so that's what we're looking at here is this PGL1 protein. What happened was that an antibody was raised against actually a different protein, and it turned out that this PGL1 protein is very antigenic, and secondary antibodies wound up recognizing it. Sorry, I shouldn't have brought that up because that's sort of beside the point. But these are bodies that localize and they surround this nucleus toward the posterior of the embryo that is going to become the germline, that will eventually become the reproductive tissue of the developing worm. But just after fertilization, what you see, this is a time lapse, is that the P granules are initially kind of throughout the embryo, and then they concentrate toward the back end of the embryo, where that germline cell will eventually arise. And it was assumed that this change from a more homogeneous distribution of P granules to this polarized concentration at the back end or the posterior end of the embryo was driven by some kind of motion that actually transported these granules through the embryo. Sorry, this is just a micrograph showing that these P granules actually eventually they become stuck to the nuclear envelope, and I'll come back to that. These are a little out of order. Okay, so here is again an embryo that has these P granules at the back, and these researchers were basically trying to figure out what the transport mechanism was that got them there. And so they started making movies of these embryos with fluorescent P granules in them. And they noticed something kind of surprising, which is that if they actually tracked the motion of individual P granules, they realized that they were not actually moving. There was no net movement of P granules from the anterior to the posterior end of the embryo. Instead, what they noticed is that P granules at the anterior end of the embryo would spontaneously just kind of get smaller and disappear, and they would see the emergence of new P granules at the back end of the embryo. So I'll play that again and I'll show you their quantification. But their conclusion is that these P granules are becoming polarized not by moving, but by spontaneously forming. And they realized that it was a condensation process. So they are spontaneously condensing out of the cytoplasm at the back end of the embryo, and they are dissolving when they're present at the front end of the embryo. And the reason that this happens, as it turns out, is there is a kinase. It's more than one reason, but there's a kinase that phosphorylates many of the proteins, and it is concentrated at the anterior end of the embryo. And in this case, phosphorylation of these proteins makes them less attracted to each other, and the dephosphorylated version of the proteins are more attracted to each other. And so in the back end of the embryo, that molecular attraction is enough to trigger condensation, and the phosphorylation in the anterior end of the embryo keeps the affinity of these proteins low enough that they don't condense. Okay, and this is probably irrelevant, but this is just the tracking that they did of individual P granules to show that there was no net movement of P granules from the anterior to the posterior end of the embryo. And so there has to be another mechanism to explain this polarity. So then they also showed by making movies, these are actually not in the embryo, but in the adult. So in this case, sorry, I'm going to go back to the slide that I showed you. So initially these P granules are just in the cytoplasm, but eventually they kind of glom onto the nuclear envelope. And so that's what you're looking at here is the P granules that have glommed onto nuclei in the germline. I haven't, yeah. I think I'm going to crash again. Okay. Maybe if it comes back, maybe I won't play the movie since it's giving me problems. But what you're seeing here in a time-lapse series is that, can you see my slides? Or have they disappeared for you?
Yeah, we can see them.
Okay, I may have to quit and restart because I'm getting a spinning beach ball. But what you can see over time-lapse is that these P granules, if you look closely at them, they undergo liquid-like behaviors. They change shape. When they come into contact, they fuse with each other. And so, and in this case, nope, it's not gonna work for me.
I want to ask, are the P granules in the cytoplasm or the nucleus?
The P granules are on the cytoplasmic side of the nucleus. So they're in the cytoplasm, but they associate with the nucleus. All right. I'm reopening my file again. All right, and I'm going to show you, I'm going to go back to this image that I showed you in kind of a not good order before. So this is an electron micrograph to just illustrate that. So the PG here is P granule, and these are nuclei. You can see the membrane around the nuclei. You can see the nucleolus, which does not have a membrane around it. And you can see that the P granules are glommed onto the surface of the nucleus, and they're actually associated with nuclear pores, which was the next point I was going to show you. And they don't have membranes around them, so they're membraneless organelles. Okay, I'll put the slides in a more appropriate order when I have a chance. Okay, so I think I'm not going to try to play the movie since that did not do well for me. Instead, I'm going to go back and talk a little bit more about nuclear pores. So I talked about pores in my last lecture in a different context. Are you seeing my screen?
Yeah, we can see it.
Okay, great. All right, so again, these are nuclear pores, and I talked about nucleocytoplasmic transport, things going in and out of this hole in the nucleus, but it's not an empty hole. So this, the nuclear pore is not itself a condensate. It is a highly, it's not a dynamic structure. So the ring of proteins that make up the nuclear pore do not, it's not liquid-like, it's very stable. But the interior of the nuclear pore is this kind of mystery material, and it's made, I talked about this at office hours if you were there. So this shows a different view of the interior of the nuclear pore. So this part of the pore, the surrounding part, you can think of as a solid, as a rigid ring. But a lot of the nuclear pore proteins, which again are sometimes just called NUPs, have these weird regions in them that have lots of short repeats in them. They're extremely phenylalanine rich. Whoops. So F is phenylalanine, and that's actually well, okay. That phenylalanine is a amino acid that forms a kind of weak pi bonds with other phenylalanines. So it's often present in these liquid-like materials within cells because it's good at forming weak bonds that are very dynamic. So a lot of these nuclear pore proteins have a whole bunch, like many copies of short repeats that are rich in phenylalanine and also glycine. And glycine is an amino acid that has no side chain, right? Just hydrogen. And that makes it extremely, makes peptide regions that have a lot of glycine in them extremely flexible because they can turn around the peptide backbone very easily. And so that is a characteristic of proteins that form condensates. That they're flexible. And so this material in the interior of the pore, which is just shown as kind of spaghetti here, is where these weird protein domains are. And no one knows exactly what this material actually looks like, but it seems like it's probably either a liquid or a gel of weakly held together proteins. And that makes it very flexible. And I mentioned that the importins and exportins can move easily through this material, and that's because their chemical surface, the surface of those proteins, is compatible. It also has a lot of phenylalanines in it, and it can kind of ooze through this kind of weird material, and it can carry other proteins with it. So this material is thought to be some kind of condensate, even though these proteins are actually brought to this spot by assembling into this ring. So this is a little bit atypical. It's a condensate within a much more rigid molecular assembly. And I don't know if I should talk about this right now. Well, I'm going to go ahead and talk about it, and it may be confusing, and I will come back and talk about it in a different way if you want. So this is these are micrographs, the fluorescence micrographs, and they're inverted. So dark here is where bright things are, and sometimes people just show micrographs that way. And what this shows, so these are cells that have in them, they've been injected with a fluorescent protein that goes into the nucleus through a nuclear localization signal. So in untreated cells, the protein is concentrated in the nuclei where it's bright, or in this case, dark. And what the researchers did here is they added a solvent called hexanediol, or in this case, cyclohexanediol. And hexanediol, I should have shown the structure, and I may have a slide that shows the structure, but it's an alcohol, so it's easily dissolved in water, but it also has a 6-carbon chain on it that makes it like hydrophobic amino acids. And so it is thought that hexanediol and similar solvents can essentially act like a detergent. It can greatly reduce the interfacial tension between 2 phases, and it can cause those phases to become mixed. And in this case, hexanediol actually makes the nuclear pore porous. So when you add hexanediol to these cells, now there's nothing keeping proteins in or out of the nucleus, and the proteins just equilibrate between the nucleus and the cytoplasm. But if you remove or wash out the hexanediol, then the nuclear pore again becomes impermeable to large proteins, and this protein can be re-imported into the nucleus and reconcentrate there. This just shows 2 different time points. So people often study these internal compartments within cells by seeing whether they're sensitive to hexanediol. And it's a little bit circular because no one knows exactly what hexanediol does, but it does seem to reduce interfacial tensions that basically will take a 2-state system, or a system that has condensates in it, and promote their mixing to form a single phase again. So they'll dissolve condensates, and it's usually reversible. Okay, I'm sort of skipping around a little bit, and I apologize for that, but this just shows in vitro, so these are in vitro now, what happens if you take one of these nuclear pore proteins that has a bunch of these phenylalanine glycine rich repeats, and you concentrate it in vitro and you cool it. It turns out that it forms a gel. And this is not a liquid. This is a substance that has some elasticity. If you poke it, it may deform, but it goes back to its original shape, and that's what makes it a gel rather than a liquid. This was an experiment to show that the phenylalanines within this tail are very important for the molecular interactions that these proteins undergo. In cells, it's not known whether the nuclear pore interior is more liquid-like or gel-like. Basically, there's a time dimension here that's important. The real question is, like, on the time scale that it takes proteins to go in and out of the nuclear pore, are these bonds breaking and rejoining, in which case it's more like a liquid, or are they stable over that kind of time frame, in which case it's better described as a gel. Okay, I feel like that was probably more confusing than was worth getting into, but now it's done, so too bad. All right, so I was talking about nuclear pores, I was talking about P granules, and it turns out that P granules, oh, I didn't want to do that. P granules are actually connected to nuclear pores. They have chemical components that some of the proteins within P granules are actually very similar to the proteins in the interior of the nuclear pore, those weird nut proteins. And the P granules themselves also seem to act as a size filter. And the way that this paper looked at that is they injected these embryos with a neutral, a dextran molecule, which is a carbohydrate molecule that doesn't interact very much with proteins. And you can buy different size dextrans. So you can buy 70 kilodalton dextrans or 155 kilodalton dextrans. And what's shown here is that the 70 kilodalton dextrans are somewhat excluded from these condensates, these P granule condensates. And the larger ones are even more effectively excluded. But small dextrans, 10 kilodalton or 40 kilodalton sized sugar molecules can co-occupy these regions. And that's very similar to the behavior of the nuclear pore interior. As I told you, small things like under about 40 kilodaltons can go through the nuclear pore, but bigger things are excluded. And so this paper concluded that these P granules actually have properties that are similar to the interior of the nuclear pore. And they also dissolve in hexanediol. I think that this movie shows this better than the image I showed before. So this is actually the reproductive structure of a worm. And here you can see that these are nuclei, and each nucleus is surrounded by these P granules. And if you take this tissue and you add hexanediol to it, those P granules just blow apart, poof, because the proteins become solvated. And then if you take the hexanediol away, the P granules will reassemble. So it's reversible. Okay, so this paper, this is just a summary of what I've already told you, is that this interior of the nuclear pore acts as a size exclusion barrier to macromolecules, and it seems that the P granule may actually just extend that environment to the exterior of the nuclei. Why it does that, we still don't know. We know that P granules are important for establishment of the germline, but we don't actually understand very much about how they regulate. They're thought to regulate the expression of a whole bunch of RNAs that localize to these P granules, but we don't really know why those RNAs are held in the P granules. I'm going to post a link, or it's on this slide, but this is an iBio seminar presentation by Tony Hyman, who's the senior author on the paper that I talked about, in which P granules were discovered to have these liquid-like properties and to be biomolecular condensates. And I think if you watch this and maybe some of the other videos that are part of this series, it will help you to hear a different way of describing the same phenomena that may help you if my description isn't working very well for you.
Sorry,
Yeah.
I have a question, Abby.
Yeah.
By virtue of the fact that the P granules kind of like an extension of the nuclear pore, does that mean that importans are also able to go within to, like, kind of surpass it in order to enter the nucleus?
Yeah, they must, because there's a lot of traffic into and out of those nuclei, even though they have P granules associated with many of their pores.
Mm-hmm. And then because, I mean, I'm guessing like the P granules, they also have a lot of like lysine repetitive chains.
Uh-huh.
Okay, I just wondered, thank you.
Okay, so as usual, like this hasn't gone as efficiently as I would have liked, and I don't want to keep you too much after the hour, but I want to talk about I want to talk about this paper. This is a very, okay, so I'm going to carry on and go a little bit longer. If you want to sign off, fine, you can watch this at your own convenience. But I want to talk a little bit about ways that people study this phenomenon and some of the unifying principles that we now think about as driving this phenomenon within cells. So so the examples that I've shown you so far of phase separation to form different liquid phases were all within cells, but people are also doing a lot of work to study this phenomenon in vitro. Some proteins like BSA, you can dissolve at very high concentration and they will never condense. They'll never form a separate phase. And they've actually evolved to be incredibly soluble. I mean, BSA is bovine serum albumin. It's at very high concentration in serum. And so it's basically evolved to not undergo condensation. But other proteins within cells, especially a lot of RNA binding proteins, have properties that make them tend to form these droplets if you put them at high enough concentration or if you cool them down. So this is an experiment where you take a protein solution at high concentration and you cool it down, and then you heat it up and you cool it down again. And what you can see is this protein is turning the liquid cloudy. And if you look at it at high magnification, you can actually see that the cloudiness is due to the formation of these little droplets. The reason it looks cloudy is because each of these little droplets acts as a lens, because there's a different refractive index inside and outside the droplet. That's why you can see it by phase microscopy. And so as light goes through this solution that's full of little droplets, it bounces around off the surface of all these droplets where there's a change in refractive index. And that makes it look cloudy. Over time, you may actually see it, undergo a more complete separation like this, although this would probably not happen in this case because the separation that you're seeing between the oil and the water is driven in part by the different densities buoyant densities of water and oil. So oil wants to rise above the water. In this case, these two, the condensed phase within the droplet and the dilute phase outside the droplet may not be so different in density. So they're probably not going to separate under gravity to form a heavier and a lighter phase, because they're not actually that much heavier or lighter than each other. Okay, so that's what I just said, that the cloudiness is due to the bouncing of light off the surfaces. And then this paper is a really amazing paper. It is, it's a complicated paper, and every time I read it, I kind of learn something new from it, and I've read it now dozens of times. But this is a paper that was published in 2012 that highlighted the role of multivalency, which is a term you may not have encountered before, in driving intracellular phase separation. So what is multivalency? So valency is basically binding. So a multivalent protein or macromolecule can simultaneously bind to multiple other partners. And that is a property of many proteins within cells, and also nucleic acids. So what these authors set out to look at was based on this hunch that they had. They noticed that a number of the proteins that seem to localize to different kinds of bodies within cells, that these bodies within cells seem to be enriched for proteins that have multiple sort of modules that can bind to other things. Sometimes some of these modules will bind to RNA. So this RRM module is an RNA binding module, but most of these little modules that they're looking at are interaction domains for other proteins. And so these proteins are basically chains of units that can interact with other proteins, and that's what makes them multivalent. So these authors actually were aware of some work from chemists going back to the mid-20th century that suggested that multivalency is a property that can drive this kind of phase separation. And these authors were particularly interested in this system. These are proteins that exist within special cells in the kidney called podocytes, and their function, that they interact with the membrane, but their function is to catalyze very dense networks of actin at the surface, and that those networks actually act as a physical filter that is part of the kidney's filtering system. And so they were really, I know this is a lot of extraneous information, but they were really interested in these proteins which have multiple domains that can interact with each other. So they hypothesized that might be driving phase separation within the cell. And so they looked at this by taking these interaction domains and making kind of artificial proteins where they just took those domains and put flexible amino acid chains between them, which are called linkers. And so they have these strings of proteins, and they can make different versions of these that have different numbers of binding domains. So this square domain binds to this kind of oval domain with weak affinity, but by making a chain of them, you can create multiple interactions between these two kind of artificial proteins. And what they looked at is how that valency contributes to phase separation. So I showed you how you can look at phase separation in a test tube. You can see it as this cloudiness, or if you use a fluorescent protein, you can see that those droplets become fluorescent. And they were able to create a system in vitro where you could get phase separation of these artificial proteins. And what they showed is that it's highly concentration dependent, and it also, so the higher the concentration of these proteins, the more likely they are to phase separate. And as you increase the valency, in other words, as you chain together in one molecule more of these binding domains, they will undergo phase separation at lower concentration. So, and if they're, at the point where you have 4 to 6 copies of each of these binding domains, these proteins will phase separate at concentrations very similar to the concentration that they're at in the cell. So, so I've mentioned a few properties of molecules that tend to undergo phase separation within cells to form liquid-like compartments. One of them is that they tend to be highly flexible proteins, like those FG repeats with the glycines. That makes them conformationally flexible. And I'll explain in a second why that's relevant. I've just explained that multivalency, the ability to bind simultaneously to multiple partners, whether they're protein or nucleic acid partners, is a property that drives phase separation. And proteins in particular that tend to phase separate often are kind of near the limit of solubility in water. They're not like oily, but they don't like they have a high proportion of hydrophobic residues, and so they are less attracted to water than other proteins. And the conformational flexibility is important because so the way that I think about this, and this is maybe not the most rigorous way to think about it, you have these large molecules like proteins and nucleic acids, and they can be kind of by themselves, just surrounded by water. They can be bound to one or two partners, or they can come together to form a much more extensive network, which is a phase-separated organelle. And when they come together to form that kind of phase-separated body, as I've already mentioned, that is a reduction in entropy, right? They're more concentrated within the cell. Just kind of intuitively, you know that there has to be some cost of taking things that are spread out everywhere and putting them all in a smaller space. And that entropy is balanced by the energy of binding between the different regions on the proteins or nucleic acids that bind to each other. So those binding interactions give, they offset the entropic cost of bringing those things together. And if the proteins are very flexible, that sort of maximizes the number of different partners that they can simultaneously bind to. And so proteins that are flexible and have lots of binding sites for other proteins will undergo this kind of phase separation readily. And those interactions also tend to be weak. And that seems kind of counterintuitive, but I've already described the fact that these are liquid-like assemblies of molecules. And in order to have that liquid-like property, each of those bonds has to be weak enough that they're kind of breaking and rejoining all the time. So I think that's all I'm going to say about this because I've already gone over. I will maybe just mention in passing that there's a lot of interest in this phenomenon of phase separation in the field of neurodegeneration. Because one thing that can happen when you concentrate molecules within a condensed phase is if it gets too concentrated, or if there's a mutation that makes the protein maybe a little bit less, that makes it prone to aggregation, what can happen is that this concentration of the protein can drive, an aberrant aggregation process. Sometimes it results in the formation of what people call amyloids, but sometimes it just results in kind of a irreversible, kind of denatured clump of protein. You can think of it as like cooking egg white. And those kinds of tangled clumps of protein are associated with a number of neurodegenerative diseases, particularly dementia, like Alzheimer's is associated with these bodies in the brain that are actually formed of proteins that undergo this kind of phase separation as part of their normal biology. And it's not yet clear whether the aggregates that are associated with like Alzheimer's disease or Lewy body dementia, whether the aggregates are sort of a cause of the disease or a consequence of the disease. But they are proteins that are naturally phase separating proteins, but if they're mutated or if the cellular environment becomes less favorable, like if it becomes too oxidizing, for example, they can undergo enough chemical change that these aggregates lose their liquid-like properties and become irreversibly kind of solid. All right, and I think that's all I'm going to say. I know this is a confusing topic, and I will give you some references to watch and read if you want a different way of hearing about this topic that may help you understand some of what I talked about today. And yeah, there's a bunch of questions on the chat. Let's see.
Someone had a question I wasn't able to answer where I think they said because the nucleus is at the posterior end of the embryo, so
Ah.
do the P granules concentrate at the posterior pole of the embryo?
That's a good question, but I didn't make that clear. No, the concentration of the P granules at the posterior end of the C. elegans embryo actually happens independently of the fact that the germ nucleus is going to wind up there. They're two separate processes, but because the P granules actually concentrate at the posterior end of the embryo before they associate with the nuclei. So they are undergoing just a spatial concentration within the whole body of the embryo. And there's a germline nucleus that is eventually going to be born in the posterior of the embryo. And at some point, I actually don't know exactly when in development, the P granules will glom onto that nucleus, but they get there separately. Someone was having trouble hearing, which is too late for me to do anything about. Sorry about that. I think there's just some kind of incompatibility between playing videos and Zoom that sometimes causes it to crash. So sorry about that, but I really like using the videos because I think they're much easier to understand often than still images. So I want to stop using them, but today was a little bit more problematic than usual. All right, and that's also why I've been editing some of these Zoom recordings, because when I go and look at them, I realize like the videos that I played didn't actually play on the recording, and so I spliced in the original video, and it takes me some time. So I'm also trying to provide links, but for the ones that I'm actually playing and narrating while I'm talking about, I try to embed them within the talk. All right, thanks you guys. You can see people are closing out their screens, so I'm going to do the same, and have a good day, and I'll see you guys on Friday. And GSIs, do you want to meet by Zoom, or you want to?
Yeah, I think that we should meet on a separate Zoom.
Okay, I'll send out a link.
Okay, yeah, maybe like in 5 minutes so I can,
Sounds good.
Okay, sounds good. Thank you.
Bye.